So today I'm going to be talking about the effects of omega-3 fatty acids on heart attack remodeling, which was my first article, and my second article, which was the effects of an omega-3 fatty acid, EPA, or ethyl eicosapentaenoic acid, or omega-6 fatty acids, GLA, which stands for ethyl gamma-linoleic acid, or the other omega-6 fatty acid, AA, which is arachidonic acid, on rats. And those rats had been injected with a substance called interleukins that caused stress and anxiety response. So it studies the effects of those different types of omega fatty acids on the rats. So my first article was about heart attack remodeling, and if you don't remember what that is or don't know what it is, basically when you have a heart attack, your heart goes through two different types of remodeling: electrical remodeling, which causes a permanent irregular heartbeat and can eventually lead to a cardiac condition, or structural remodeling, or fibrosis, which is basically just buildup of scar tissue in the heart, and that causes the heart to not be as flexible and not be able to pump as much blood volume with every beat. So the study was done on dogs, and they were orally given daily PUFAs, or polyunsaturated fatty acids. They had been, one group, or I think all the groups, had been induced with electrical and structural remodeling by what is called atrial and ventricular tachypacing, which is basically just electric shocks to the heart to cause an irregular heartbeat and to cause fibrosis. So what was found was that the group that was given omega-3 fatty acids daily had significantly less structural remodeling, or less fibrosis, less scar tissue buildup, than the group without the omega-3 fatty acids. And this was thought to be because of the inhibition of MAPKs, mitogen-activated protein kinases. So somehow the omega-3 fatty acids inhibited the mitogen-activated protein kinase, specifically the C activity. This is basically a chain of events. It's a second messenger, and if you've had AP Bio, you know that a second messenger is secreted in response to a hormone released by the endocrine system. So basically, it's a messenger inside the cell that brings about a certain response. In this case, MAPKs do multiple things, but in this case, they direct pro-inflammatory cytokines. So when they're released, they cause inflammation, and it is thought that the omega-3 double bond somehow inhibits MAPKs. My second article was on the different influences of omega-3 and omega-6 fatty acids on both inflammatory response, and the stress-anxiety response. So basically, the rats were injected with interleukins, which cause stress, anxiety, and inflammation. And one group was given EPA, ethyl eicosapentaenoic acid, which is an omega-3, and then another group was given GLA, and yet another group was given AA, omega-6. And so the results of the study were displayed here. The EPA, or the omega-3, reduced the inflammatory sickness due to interleukins. And interleukins, I should probably explain that. They're another signaling molecule, and they increase the secretion of prostaglandin, which causes that inflammatory. And I'll go over more of that a little bit later. But they reduce the inflammatory sickness. They reduce the stress response. The GLA reduced the inflammatory sickness, but they had no effect on the stress response. And the AA, the arachidonic acid, had no effect on the inflammatory sickness or stress response due to the interleukins. However, they did increase the secretion of corticosterone, which caused its own inflammatory and stress responses. So I'll go a little bit over those right now. Prostaglandin, it's an eicosanoid, and it can have different effects based on the receptors that it's binding to, but here it does cause inflammation. So when prostaglandin was secreted, it causes inflammation. The ethyl EPA and GLA, however, decreased prostaglandin secretion and increased anti-inflammatory cytokine secretion, so they both reduced inflammatory sickness. However, the AA, although it didn't have an effect on the interleukin-caused sickness, it did increase concentration of corticosterone. The corticosterone increased anxiety behavior, the basal inflammatory response, and raised corticosterone concentrations. Corticosterone doesn't really have a big role in humans. It's another hormone. However, it affects small animals, the stress in small animals such as rats, quite a bit. It's produced in the adrenal glands, and it is involved in a regulation of energy, immune reactions, and stress responses. So basically, the immune reactions would be an increase in inflammation, which would cause those B cells and T cells to come to the site. And the stress response, obviously, is stress. So they would be more stressed if more corticosterone was increased into the body. Now, although GLA and AA are both omega-6 fatty acids, as you could see back here, they had different effects. They had drastically different effects on the rats. Now, GLA is actually a precursor of AA, but it inhibits inflammation. Now, most humans regularly consume the omega-6 fatty acid, linoleic acid, which is the most common omega-6 found in our diets, and that's converted into GLA, which is a more useful form. But when you consume too much AA is the most consumed omega-6, it promotes inflammation and prevents that conversion of linoleic acid into GLA, which is why you need a balance. And here's the structure of linoleic acid and the structure of GLA and the structure of AA right here. So where to go from here? Obviously, the effects of PUFAs on hormones are a big indicator in the effect that they're going to have on humans. So we can study the effects of omega-3s and omega-6s on the endocrine system. Also, more research into mitogen-activated protein kinases, how the omega-3s specifically inhibit them. We mentioned briefly back up there the double bond. The double bond somehow inhibits it, but we could do some more detailed research into that. And there's our reference. Thank you for listening.